Hello students, so in this question in part A, lactose is present and glucose is absent. So then in that scenario, there is this lac operon. So lac operon has structural genes, regulatory gene and a promoter. So there are these ZYA, these are the structural genes. This is the promoter. Here there is an operator and this regulatory gene produces lac repressor. So what happens is that this Z produces beta galactosidase enzyme which breaks down lactose into glucose and galactose. Y produces beta galactosidase permease enzyme which transport lactose and its analogs into the cell, and A